Okay. So students, if you could see in this video, you could see the colorimeter instrument in front of you. So if you see this instrument, before you actually start using a particular instrument, it is very important to use the correct switches. So in that case, make sure that the main switch of this particular instrument is connected to a wire. Okay, so make sure that you have switched on that, and understand that only after switching on the main switch you are supposed to switch on the switch which is present in the particular instrument. So this is universal for all the instruments which you are using. So please understand that. So if you are going to switching on this one, and after that if you are switching on the main switch, there may be an increase in the voltage, and that will affect the working of the instrument. So please understand this. I am telling you again, whenever you are using any instrument in our laboratory, you must make sure that you are going to switch on the main switch of the wire where electricity comes, and after that only you are supposed to switch on the switch which is present in the particular instrument. So if you are doing mistakes with this, that is going to affect the duration of the specific instrument which you are using. So please understand that. So these things you must remember. So you could see this is the view of colorimeter, and when we switch on, you could see a display over there wherein it is displayed as CAL. CAL means that it is calibration. So we need to calibrate a colorimeter before we start. Okay, so we need to calibrate a colorimeter before we start. Without calibration you cannot use this particular instrument. Okay, so before calibrating, let me just tell you what is the purpose of using colorimeter. Let me just tell you that briefly, and after that we will move on to the calibration. So basically in the theory also we have learnt about the electromagnetic radiation, about the different spectroscopy also. Likewise in colorimeter also we use colorimeter for estimation. Estimation in the sense quantification. What do you mean by quantification? We are going to quantify, or we are going to estimate the amount of a particular biomolecule present in a solution. Say for example, I have a solution in a test tube and I want to measure how much amount of glucose present in it. So in order to identify how much amount of glucose present in it, I will be using a colorimeter. Okay, so please understand, this instrument is basically used for quantification, for estimation. Okay, quantify biomolecules, how much amount of glucose is present in a solution, how much amount of amino acid is present in a solution, how much amount of fatty acid is present in a solution. So these biomolecules can be estimated quantitatively using colorimeter. So this is the basic purpose of using this colorimeter. Speaking about the different parts of colorimeter, let me tell you that briefly. So inside this colorimeter, there are many parts available. Maybe the first important part is going to be light source. The second one is going to be the slit. The third one is going to be the condensing lens, and the fourth one will be the filter. The fifth one is going to be sample holder, and the sixth one is photocell, followed by the last part of this colorimeter is galvanometer. Okay, so these are the different parts which are available inside this. So inside this colorimeter, please understand that these parts are assembled in such a way that this colorimeter is working properly. Okay, so in the theory classes we have learnt about electromagnetic radiation. So we understand that sunlight is the ultimate source of light, wherein with the differences in their wavelength we are able to get different colors. So please understand, sunlight is the ultimate source of light, and with the differences we talked about the wavelength in the theory class, that how does a wavelength goes with. Okay, and based on its differences in its intensity and amplitude, the color of that particular sample is going to differ. So please understand how colors are produced. It is actually produced based on the differences in the wavelength. So we have a light source inside this colorimeter, okay, from which we can select different wavelength to produce different colors. So that is the basic idea. So we have the light source, and we also have the slit and condensing lens, and these two slit and condensing lens makes the light rays parallel. Okay, so inside this colorimeter we have a slit and condensing lens that makes the light source parallel. Okay, and the fourth one is going to be filter, or which is also called as wavelength selector. which is called as filter or wavelength selector, that you could see here. In this colorimeter, you could see a button which is named as filter. Okay, so this filter is also known as wavelength selector. So the specific wavelength will be selected using this filter. So please understand, in the theory class we have explained about the electromagnetic radiation. So here with colorimeter we can only use visible range. Okay, so say for example, before rain we could see the 7 colors, that is VIBGR. So this VIBGR comes under visible spectrum. Okay, so we learned about that different spectrums which are available in electromagnetic radiation. Say for example, ultraviolet radiation, visible light. Okay, you have radio waves, you have microwaves and so on. Infrared rays, as I told you, infrared rays is commonly used for changing your or navigating the channels in your television. So something like that, different wavelength is used for different applications. So here please understand, the filter which we use in here is actually in the range of visible spectrum. Okay, it is going to be visible range. So the visible range always lies between 400 to 700 nanometer. So please understand this. The visible range always lies between 400 to 700 nanometer. So below 400 nanometer, what is that? It is going to be ultraviolet range, which is not available in this colorimeter. Please understand that. Okay, we cannot use ultraviolet radiation in colorimeter. We can use only visible range. So that visible range actually ranges from 400 to 700 nanometer. As you could see in this particular colorimeter, the range itself, it has been given 400 to 700 nanometer, it has been given. So please understand that. So we can use only the visible range. And now you could see a number, 2 numbers which are actually printed there. It is 55. So please understand, what does it mean? 55 means it is 550 nanometer. Okay, it is 550 nanometer. So let me scroll it. You could see one number here. Now it is going to be 520 nanometer. Okay, again let me scroll it. So this is going to be 490 nanometer. And this is going to be 470 nanometer. So something like that, you can keep on scrolling it. See, this is 430 nanometer. But then please understand, it is always between 400 to 700 nanometer. So now it is 430. See, again now it is going with 700. Okay, so the visible range is going to be between 400 to 700 nanometer, and this part is called as filter or wavelength selector. Okay, so this part is called as filter or wavelength selector. And very near to this filter or wavelength selector, you could see a hole. I hope you are able to see that hole, which is very near to the filter. So that hole, what is the purpose of this hole which is there? So please understand, that is going to be sample holder. Okay, so this is the particular hole, this is the particular provision which is used for holding the sample which we want to quantify. I have a biological solution, so that biological solution will be actually measured, which will be kept in this particular sample holder. So this part is called as the wavelength selector or filter, and this part is going to be the sample holder, and here you could see the display. Okay, alright. In addition to this, we also have a button which is here, present here, you could see. Okay, very near to the display we have a button here which is set 0. Okay, we have a button which is called as set 0. So what is the purpose of having this particular button? We actually use this button to set the blank. Okay, we actually use this particular button to set the blank. A blank solution is going to be simply a colored solution. Please understand, a blank solution is simply a colored solution which we use in colorimeter. Okay, so by using the blank we have to set 0 in display. Okay, so after keeping a particular solution we actually set 0. That solution which we actually keep initially during the beginning of an experiment is going to be blank, and that blank will be always 0. We have to set 0. So that is going to be set 0 button. So these are the different buttons which are available in colorimeter. So please understand, we have a filter, we have a sample holder, we have the display, and we have the 0 setting button. So these four different parts which are available external as far as the colorimeter is concerned. Inside this, as I told you, you remember the different parts which I have told you. Let me just tell you that once again. You have a light source, you have a slit, you have a condensing lens, you have a filter or wavelength selector, and we have a sample holder, and followed by the photocell and galvanometer. So we will be speaking in detail about the working mechanism of colorimeter during our practical class. Thank you.